Hello everyone, my name is Taryn Dunivant and I am a master's student at the University of Kansas in the Department of Ecology and Evolutionary Biology. And I am in the Heilman lab where I am doing a gene assay to study the bilateral symmetry in this little flower called Mimulus lewisii. I am also an outreach fellow for the National Center for Science Education for the year of 2019. And so I just want to show you some of the different projects and things that I do day to day as a grad student and as a fellow for NCSE. So besides science, I love music. So I collect vinyl records. I listen to a lot of jazz and soul, but I love all sorts of other genres as well. I love live music. I love recorded live music, so that's a lot of the vinyl that I listen to in the house. I also love to garden. I love collecting house plants. I have them spread out throughout my house in all sorts of different varieties, and it's always fun to find something that I don't have. And I love being creative, and I love creating things and making little art projects, and I think that this is actually very helpful and useful when doing science. You have to create experiments and adjust, and so I think that my love for creativity has actually very much helped me in science. My first assignment as a fellow with NCSE was to create an outreach activity called Climate Change in My Community. And this activity needed to use some sort of a data set related to my community. So I went onto the NOAA website and I was able to find the annual precipitation of Kansas from 1895 all the way to 2018. And so this is public data, and anyone can go onto the NOAA website and they can find the annual precipitation for any state that they like, or you can even find the annual precipitation for counties, and you can find temperature as well. And so I use this data to make a big interactive graph. And this was all related to tree specimens. So I got in touch with a local arborist here in Lawrence who donated some tree specimens to me. And I took them and I gave them a nice sand, and then I polished them by spraying them with clear spray paint. So it gave them this great finish, and it also helped for the rings to pop, so it was easier for people to count the rings. Later on, I ended up adding these rhinestone stickers, so people that maybe had even trouble reading, or younger kids who had trouble reading the tree rings could more easily count them and see the difference in the growth ring size. And so along with all this, I was also able to then show people some tree identification, and so the difference between native and invasive species, as well as how trees actually grow and the anatomy behind that, and then altogether how this correlates with the precipitation. And so in years where there is not a lot of rain and it's more dry, we're going to have thinner growth rings, and then in years where it's really wet, we're going to have wider growth rings because trees are able to put on more growth. And so we can string all this together, and we can use the study of dendroclimatology to study climate change and long periods of change in an environment. And so in trees, there's all these cool other different environmental events that get recorded in the trees and different climatic conditions. So these are all different aspects that I had integrated into the activity. And so I will show you actually how I made this big interactive graph. It's a beautiful day here in Lawrence, Kansas, and I just picked up my poster for the activity that I'm creating using climate change data from my community. So I'm gonna go home and I'm going to build the rest of the activity. To make the bars for the graph, I cut a bunch of strips, like this. Then I measure each strip so that it correlates to the inches of rain per year for that year, and then I write the year and the amount of rainfall on the back of each strip. So I finished creating the bar graph and all the strips. Now I gotta add magnets to the back so that they'll stick. So overall, the activity was a success. The kids loved helping me build the graph. I'd say, hey, do you want to help me build my graph? And they'd be like, yeah. And then we'd build the graph together, so I was able to teach kids what a graph is, how you read a graph, how you read times, and looking at the different bars, and so you can see the variability in precipitation over these periods of times, and that was really connecting with the kids. And my eye candy here, my tree specimens, really drew people in. So the shiny coating, people are like, hey, what you got over there? And so kids and adults loved looking at the insides of the trees. And I feel that I was really able to convey the importance of trees and how climate change may be affecting trees in the future. And so that was great. I was very pleased that the work that I put into the activity was reciprocated in the community. Good day, everyone. Happy Earth Day. Today I'm here at South Park, Lawrence, Kansas, and there's a big festival going on. There's a parade, food trucks, lots of booths set up. Even the KU Natural History Museum has a pop-up museum truck. So I'm so excited today to do my climate change activity, which is connecting variable precipitation over Kansas along with how that affects growth rings. So I'm really excited to share this with the kids and the families of Lawrence, and let's go check it out.
Just like to spin it. Get some information about that.
So it's a Sunday, but I'm here still getting work done. I am transplanting my seedlings and separating them out so that they have a chance to have more soil and grow bigger. So I'm here at the greenhouse at KU doing that, and I'll give you a quick little tour. All the different pot sizes that you might need. Soil! Here's my plants I'm working on. So these are the seedlings that, the seeds that I germinated, and now they're getting transplanted. So each one has their own little home. And here's a look at the greenhouse. Different research projects going on, teaching collections, composting, and whatnot. So I'm gonna finish transplanting these and get them back into the growth chambers. So I just finished transplanting 70 seedlings, 35 are wild type, and 35 are transformed transgenic plants. So I'm going to grow them up and see if I see any mutant phenotypes. Good morning and good day, everyone. I'm just finishing up my coffee. It's almost 8:30. I have my first meeting at 9, so I gotta get out of the house and take my walk over to campus, which is one of my favorite parts of my morning, is getting my mind right and getting ready before I get on campus. So today's Tuesday. I have a very busy day. I have discussion, I have seminar today, a couple meetings, and I also have to put in some lab hours. So I'm gonna bring you with me and show you what it's like in a normal day of a grad student here at the University of Kansas. Okay, so now we're here in my office on the 7th floor. I had my meeting with Dr. Atkinson, who's my advisor in the paleobotany lab, and we were just going over what I did last week and what I'm going to do this week to fill my hours for my research assistantship. That is how I earn my stipend, and that is also how I earn my tuition. But I've been in here finishing reading a paper for my discussion group, and now that that's done, I'm gonna head down to the paleobotany lab and put in a couple hours, and so I will show you guys how to make a fossil peel and give you a quick little tour as well. I love having my office up here on the 7th floor. I get this beautiful view of the south end of Lawrence, Kansas. There's been thunderstorms rolling in and out of the city. As you can see with these gray skies, it's supposed to start thundering later in the afternoon, so we might have to deal with that as we walk across campus to some of the different things that I gotta do today. So no one's really in my office right now. It's just me. I think maybe next semester I might have someone joining me. I love having this private space, but I got some of my outreach supplies up here from the NCSE poster I've made. Of course, I gotta have some plants in here, keep me healthy, some pictures of friends and family, but it's a great quiet place for me to get a lot of my work done. Good afternoon, everyone. It's a Saturday, and today I'm headed over to the Lawrence Public Library to give a talk. And it's a talk that I was invited to give, and I'm so excited. I will be talking today about growth rings, and I actually took the activity that I created for NCSE, the activity of climate change in my community, and I turned it into a 10-minute talk. So today I'll be giving that talk about growth rings and all the different environmental conditions that get recorded in trees and that you can use to understand and reconstruct past climates to help us understand the future and see what's going on. And I'm so excited. And after that, I'm gonna have a table set up with the activity as well, so people can come see real specimens, we can put together the graph, and I'll let you know how it goes. It's a record. They're like little secretaries that are documenting what the environment has been like in the past and currently, and it will also in the future. So first, let's start with some basic tree anatomy. So trees have two main types of tissues. They have phloem, which is moving the sugars up and down the tree, which is their food source. And they also have xylem, which is piping that's moving the water from the roots through the tree and all the way up through the leaves. So let's all thank trees, and thank you for listening. Okay, so you had a day in the life with me. One day I had all these things to do, but you actually haven't been here in the lab with me. So I wanted to give you a little bit of a tour of the lab, and I actually have to finish a task, so you can help me finish my task. So this is my lab. I am in the Heilman lab, and everyone has their own little workspace here in the lab, the fume hood. This is my workspace over here, right next to the centrifuge. We also have this room in here where we can do things like run gels and then even take pictures of the gels. So a couple days ago, I did some DNA extractions from my plants, the Mimulus lewisii, and I am screening to make sure that the vector that we were trying to use to transform the plant actually got implemented into the DNA. So I did some DNA extractions and I just did some PCR. So it's been running here in the machine, but it's done. And tomorrow morning I will run some gels to see if that piece of DNA was actually inserted into the plant. So now that my PCR is done, I just turned off the machine. We're gonna open this up, lift it up. So here are my samples. So we're gonna take these out and we're gonna put them in one of these little cells for the PCR tubes. And tomorrow I'm gonna run a gel. So since I'm not doing it tomorrow, we're gonna take these samples and we're gonna put them in the refrigerator. So in the refrigerator, we keep lots of different enzymes and all these different things that need to be kept at a particular temperature to stay stable. So while we're in the lab, I thought it would be a good opportunity to talk to you about my research and some of the struggles that I've had here in grad school with research. And so coming here to KU, I wanted to be a paleobotanist, and so that was the original master's thesis. I was describing a new species of fossil. However, while doing this research, I realized that it really was not the right field for me and that it was not something that I would want to continue on with in the future as a career. And so I sat with myself and I realized that I needed to make a change and that I missed working with plants and touching plants and holding plants and working with extant taxa. So I decided to turn to plant genetics, and I was happy that a new advisor took me on and had an amazing project ready to go for me. So now what I'm doing is that I'm looking at this plant, the purple monkey flower, Mimulus lewisii, and I am doing a gene assay related to the floral morphology. So I am looking at the bilateral symmetry of the flower and which genes are responsible. And so what I want to share with you is that to not be scared of change and to try things. I thought I really wanted to be a paleobotanist, and I would have not have known that that was not the right field for me until I actually tried it. Hi everyone, I wanted to share with you the new kit that I received in the mail from NCSE, and I'm so excited. So this is the May kit. It is the archaeology kit, and I got a bonus with my kit. I got this awesome t-shirt. So science makes us strong, and I agree. I know that science has made me such a stronger, more powerful and intellectual woman, and I'm so happy to be a scientist. And that's why I'm here sharing science with you, because I also help to empower you. And so the kit itself is small, which is great because going out and doing science outreach, it's a lot of travel and bringing your own supplies and setup, and so having something that's very easy to travel around with and that is very accessible is such a bonus. So this kit has three main focuses: wolf domestication, the domestication and cultivation of agriculture, and an archaeological dig. And so when you open it up, there is this fun game that goes with the wolf domestication, and this is a choose your own adventure. So you're going to go through different generations of wolf pups until you get to a particular breed of dog. And this activity shows that the evolution of dogs has been based more on behavioral traits rather than physical traits. Then there's a fun matching game. Do you remember when you used to play flip the cards and you try to find the matching pair? Well, that's what this game is, and you're trying to find the wild version of the animal or the plant, and then the domesticated, cultivated version that we now use in agriculture. So for example, this is what wild cotton used to look like, but this is what the domesticated version of cotton looks like. Or with a rose. So a wild rose actually only has 5 petals, but the cultivated rose has whorls and whorls of petals and actually has even lost some of their reproductive parts. So even with animals, the wild cow has these horns, and the domesticated cow has lost those horns and has gotten larger for meat and milk production. So this is just showing how humans have taken these wild organisms and cultivated to help them build the society that we have now. The last part, the archaeological dig, is this really fun tray here. So this would be like an archaeological site, and these are the little tools to help them dig, and inside are some different bones. So these are some human bones, some pieces of tools that humans would have created, and then some bones. This one here to me looks like a tusk. Oh. And so this is to show that humans have been using tools and domesticating animals for a very long period of time. And so I'm gonna fill this tray with sand, and I got this huge bag of sand, which I'm actually really excited about because I did need sand to do my Rising Tides activity, so now I have a good amount of it to do other activities for NCSE as well. So I'm going to fill the tray with sand, and then I'm going to bury these different artifacts at different levels, and it'll represent all of these things happening in a relatively the same time period in the same location. So tomorrow morning I'll be getting up bright and early, and I'll be headed over to the Lawrence Farmers Market, where I'll be setting up my booth, and I'm going to do this activity, and I hope that it goes over well with the community, and I will share with you my experience and how that all goes. Good morning, everyone. It's about 6:30 and I'm about to head out to the farmers market. It's raining outside, so that will be a fun setup, but once I'm set up, I'll be nice and dry and able to share science. So let's go do it. So I'm still talking to a decent amount of people here at the farmers market, but not as busy as last week, I think, because the weather's pretty bad, but people are learning a lot and really enjoying the matching game. So today at the farmers market, I had a really great time. I was able to talk to about 60 people, so only about half as much as last week. But this time, because it wasn't as crowded, I was actually able to spend a lot more one-on-one time with families and different individuals. So I was able to go through full games with families, and parents were playing along with their kids, and siblings were playing together, and so that was great. And everyone really wanted to hear the information that I had to share, and everything was really well received, and I got many thank yous for being there. So even though I want to be there and it's rewarding just sharing the science, it's also nice to get some gratitude from the community. And so overall, despite the rainy weather, I had a great time, and it's always fun sharing science with people. I'm really enjoying being an outreach fellow for the National Center for Science Education. So I just wanted to share with you why science outreach is important to me and why I do science outreach. And there's pretty much 3 reasons for that. The first reason is that science is important. So So science is important to me, but I think it's also important to everyone else in the world because it leads to progression and advancement for all of us. And so as scientists, it's really important that we communicate our science and that we communicate it in a manner that everyone can comprehend. And so that's why I think that outreach is a great avenue for this. Secondly, as a kid, I got little to no science outreach. So it wasn't until I was about 26 years old that I realized that I love science and that as a woman, I could become a scientist. So I want to reach out to younger generations and show them that anyone can become a scientist and also that you can do it at any age. And so I'm a great example of that, and I want to share that with people. Thirdly, and one of the most important things to me is to share the concept of plant blindness with people. And so plant blindness is a condition that's sweeping across the globe, and what it is that it is the inability to see or notice the plants in one's own environment. So it's basically an underappreciation of plants, which really hurts me because plants are alive, and I know that plants are ubiquitous and that they're all around us, but they're still living and breathing organisms, and they do so much for us. They provide us with food and oxygen and medicines and materials and shelter and even habitats for all the other little animals that we love. So without plants, we would not be here. And so I think that it is important to share the impactfulness of plants on the planet and some education about plants. And so these are the reasons why I do outreach and why I share my science and the other science with people. And I have such a great time doing it, and I'm so happy for this avenue of the NCSE for allowing me and giving me all the tools to be better at doing science outreach.